Okay. So, hello everyone. My name is Shingo Matsumoto from Hokkaido University, Sapporo, Japan. And in my talk today, I'd like to focus more on the in vivo application side of FIP technique. Um, so I'd like to share our recent findings on the visualization of brain metabolic alteration, especially associated with retinal degeneration using parahydrogen-polarized carbon-13 pyruvate MRI. So we are currently exploring the new biomedical application of hyperpolarized carbon-13 MRI technique, and particularly non-invasive evaluation of brain metabolic alteration under the polarized stress and disease condition. And FIP is actually will suit this kind of purpose, and it's because of its very quick polarization build-up time. So today, I'd like to show one representative result in the case of vision impairment model. So vision impairment caused by the retinal degeneration is not just an eye issue. It significantly increases the risk of brain-related conditions, including cognitive impairment and depression and anxiety. And conventional anatomical MRI is limited in detecting such a psychological or functional disorder, especially when without the structural abnormality can be observed. So this motivated us to use hyperpolarized carbon-13 MRI or pyruvate metabolin to detect them. So here we prepared two different mouse models of retinal degeneration. And one is a congenital model known as RD1 mice. It's carrying a PD6B mutation, representing the chronic retinitis pigmentosa. And the other acquired and transient model of retinal degeneration is prepared by the exposure to the 10 hours fluorescence light. So we confirmed the retinal histology showed a significant decrease in the outer nuclear layer thickness in the retina of both RD1 and light exposed model mice. So to investigate the neurological abnormality induced in these model mice, we conducted the three different behavioral tests. An interesting point is that these two retinal degeneration model mice showed a distinct pattern of behavioral abnormalities. For example, in the spontaneous locomotor activity test, only RD1, this is the chronic and the congenital RD1 mouse, only show the decrease in the nighttime moving distance within the cage. In contrast, in the tail suspension test and elevated plasmid test, this light-exposed mouse model displayed the typical depression and anxiety-like disorder, um, whereas the RD1 mice show the completely opposite abnormal pattern. So before conducting the imaging study, to investigate the possible brain metabolic alteration, we measured several biochemical markers. And the brain lactate concentration level were increased in both light exposed and RD1 mice, that corresponding to the lower brain pH level. And both retinal degeneration models show the increased level of pyruvate dehydrogenase kinase 1 expression and also phosphorylated pyruvate dehydrogenase level. Whereas lactate dehydrogenase expression level remained unchanged in both models. So also the representative immunofluorescence image showed that phosphorylated PDH was present also in neuron, but predominantly in astrocyte. So maybe metabolic plaque change may be happening and mainly dominated by the astrocyte in this study. And brain metabolic alteration was further confirmed using the proton NMR-based metabolic analysis. And this is the summary NMR study, not the hyperpolarized study. And among the 42 brain metabolite, hydrophilic metabolite identified, 18 metabolites were actually significantly changed either retinal degeneration model. And this kind of clustering analysis clearly separated the the cluster of both retinal degeneration model from the healthy control mice. Then for the non-invasive imaging study, we have our carbon-13 MRI system, which can be combined with preclinical PET and SPECT scanner in our university. And so at first, we conducted that F18 labeled fluorodeoxyglucose PET imaging to investigate the brain glucose uptake. And the mean standardized uptake value of the FDG was almost about 20% lower in the brain of both light-exposed and RD1 mice compared with healthy control mice. So this brain glucose consumption rate and also the overall, it reflects the overall brain neuronal activity might be attenuated or might be decreased in the case of associated with vision impairment. So for the PIP study, we use our lab-made PIP polarizer system, and we made a trick for the proton to carbon spin order transfer, and in which the three-dimensional fluid path was placed under the very small magnetic field gradient, and just parahydrogenated precursor was flow through this fluid path, and the spin order of two hyperpolarized proton can be transferred to the labeled carbon-13 atom. And more detail of this polarizer system is available in this paper. Then we applied this hyperpolarized pyruvate MRI imaging to this retinal degeneration model. And carbon-13 level of this pyruvate solution was about 4% at the time of imaging, so it was quite challenging imaging. And this 2D brain chemical shift imaging was obtained about 25 seconds after the pyruvate injection. And a significant reduction in the metabolic plaques to the bicarbonate was commonly observed in both models. And in contrast, the metabolic plaques to the lactate was unchanged in the chronic RD1 mice, whereas it was increased by 1.5-fold in the light-exposed mouse model. So these results suggesting that this hyperpolarized carbon-13 MRI might be useful for detecting the brain metabolic alteration associated with vision impairment, and even absent of the clear morphologic abnormality. And then recently, maybe some of you know, recently, interesting paper was published in the MRM, and here, during the hyperpolarized pyruvate MRI measurement, presenting this kind of visual complex pattern to the subject, and this stimulates the visual cortex and reading the immediate increase in the hyperpolarized bicarbonate flux in the brain. So this finding suggested that since our imaging study shown the decrease in the bicarbonate flux, so maybe neural activity in the visual cortex may be attenuated in the case of vision impairment. To verify this, we conducted the immunohistochemical evaluation of brain neural activity marker CFOS expression. So CFOS is one of the immediate early genes, and which was immediately expressed when neuron was activated, and it's disappeared within a few hours. So it's serving as a marker of neural activity. So in this number P4 slice, it's containing the visual cortex region, and this red fluorescent signal you can see in the control mouse around the visual cortex region, but it's clearly disappeared in the chronic RD1 mice model. And the bottom image shows another fluorescence image of phosphorylated PDH. So this red fluorescence indicate the suppression of metabolic flux to TCA cycle, corresponding to the suppression by carbonate flux. And as you can see, the almost no suppression of TCA cycle was observed in the healthy control brain, but it was markedly suppressed in the chronic RD1 mice. So these findings suggested that maybe there is some correlation between the attenuated neuronal activity and suppression of pyruvate metabolism, so which can be observed hyperpolarized carbon-13 monoide technique. So these results were made possible through support from my collaborators and research grant. Also, we will have Envision price system maybe next June. Hopefully, no more delay of delivery. So we will be able to provide more better brain imaging result maybe in the next per meeting. Thank you very much for your attention.
All right. We have time for maybe one or two quick questions. Yes, I have one. Can you talk a little bit about quantitatively the difference in sensitivity you expect for looking at the depressed brain activity, comparing the hyperpolarized pyruvate to the FDG model?
No, we try to compare the FDG and the pyruvate imaging, but still our imaging accuracy or some spatial resolution is not enough to compare the particular region of the brain. So maybe after we get the polarized system, maybe we can do such an analysis of correlation between FDG and pyruvate metabolism.
Okay, right. Thank you. I'm not seeing any other questions, so let's thank our speaker once again.